Welcome to Lecture 13, Qualities of Scientific Evidence. Have you ever heard the phrase, studies show? What does it mean when we say studies show? Does it mean anything? Is it good that studies show? Is it bad? In order to make sense of the information, you need to know how to assess an individual study. First of all, we look at what kind of study is it? Is it an experimental study or is it an observational study? And when we look at the experimental and the observational study, we have to balance all of the evidence. Pro evidence and con evidence, evidence for my theory, evidence against my theory. In experimental studies, we look at some specific intervention. We do something to affect the world. And once we do something to affect the world, then we have to measure it. We have to measure the outcomes. And as you're doing this experiment, we have to be able to control for as many variables as possible. And ideally, only one variable will be completely isolated. So we can tell that this variable has an effect or doesn't have an effect. There are strengths of each of these different types of studies. With experimental studies, we are able to control a wide variety of variables. And hopefully we're able to control all of the variables. That would be ideal, so that we can focus in on the one that we're controlling. We're able to perform quantitative analysis of that information that we get, to see whether that information actually has an effect or not, whether our intervention that we did actually caused any effect. And we're able to perform the statistics to compare different groups. So we can compare our control group to our intervention group to another group that we're giving something totally else, something totally different. The observational studies have other strengths. We're able, through observational studies, to gather vast amounts of data. We're able to compare different groups with this data, and we're able to do it with the absolute minimal intervention. Strengths. One thing we want to look at is how large is the study. One study is still one study, and one study is subject to errors and bias. It's done by people who have biases, who have errors. We also have to look at, is this group, is it representative? Many studies are done on college freshmen because the university professors need to do some studies, need to publish in order to keep their jobs. It's very easy for them to study their college freshman class, offer some extra credit, come in and participate in the study. But is that group we're studying really representative of the general population? So we have to look at, is this group representative? We also need to look at publication bias. Publishers tend to publish studies that have positive effects and not publish studies that have the negative effects. So they have a strong tendency to publish studies that actually show the hypothesis being true, but not publish studies that show the hypothesis being not true. There's a type of studies called meta-analysis. And meta-analysis is when I'm not doing the study myself, but I collect studies from dozens and dozens of other researchers who have done the studies, and then I put all of their data together so that I have one humongous group of data, and then I run the statistics on this data. And this is a great thing to do, but unfortunately we stumble across publication bias because I find those studies through publications. And it could very easily be that garbage in and garbage out. That if the only things that were published were the positive result studies and not the negative result studies, then my meta-analysis itself is going to be skewed. Science and the scientific methods are ways for compensating for the flaws in our brains, for the flaws in our thinking, for our logical thinking errors. We need to find out really not what is true, not what we want to believe is true. We look at studies, we're looking at, was this study statistically significant? And for the most part, statistic significance says, does it have a p-value of 0.05? And I know people don't understand that, but what this means is that 1 in 20 will give you a false positive. That no matter what I'm studying, I'm going to get a false positive in 1 in 20 people I study, or 1 in 20 studies will actually have a false positive. And that means 19 were what I assumed it was going to be, and this 1 is going to sneak in there and mess up my stats. And statistics are not everything. We also have to look at the effect size. If I'm studying an illness or a test for an illness, and it has a false positive of 1 in 20. So it sounds like that if I study 20 people, one of them will test positive for that illness. And that seems appropriate, that one of them tests positive for that illness. Or if I'm studying 100 people, then 20 of them will test positive even though they actually don't have that illness. This falls under statistically significant. However, what we have to look at is the actual base rate of this. What is the effect size? So if I'm studying an illness, but the illness only occurs in 1 in 1,000 people, and yet my error rate is 1 in 20, I'm making an awful lot of mistakes with this type of error rate. So we have to look at what is the base rate, or what is the effect size. And we need to look at, was the data collected continuously, or was the data collected in a stop-and-go fashion? This is where studies of psychic abilities they used to do this study. They used to say, okay, sometimes it takes the psychic ability time to warm up, and then after a little while the psychic ability gets tired, and so it doesn't work too well. And so they would flash people cards and have them guess on what the card is going to be, and mark down every correct answer and every incorrect answer. And the law of large numbers says there are going to be strings of correct answers, and there's going to be strings of incorrect answers. And what these researchers were doing was saying, okay, psychic ability takes some time to warm up, so we're going to throw out all the negative answers before that string of positive answers started. And then psychic ability actually wears off for a while. We get tired. And so we're going to throw out all those incorrect answers after the string. And so the researchers were starting to come up with strings of, yes, psychic ability really does exist, because they were doing their data collection in a stop-and-go fashion. Surveys are another big challenge. There's typically problems with a survey. Are all of the people who are being surveyed actually being counted? Are we weeding out? Are we sending the survey to a representative population? Are we blinding the people about what the survey is actually about? What are we looking for? Are there leading questions in the survey? Is the survey coming from a political organization that obviously has a political affiliation? And some of the questions in there are also political questions. You can tell how the survey wants you to answer. So are there leading questions in this? There's lots of problems with lots of studies, which means is why we need our critical thinking abilities. Why we need to be able to publish and get other people's opinions. That one study is simply one study. One study doesn't tell us a whole lot. Well, one study gives us a place to start. And then we let other people reproduce our studies, come up with entirely new studies to test our hypothesis. And finally, with any luck, we'll have a consensus of agreement that yes, this is true, or no, this is not true, through consensus of opinion, through a lot of these people studying. When the data source becomes large enough, when enough people have looked at the statistics and said, yes, this is good. In our next lecture, we're going to start talking about pseudoscience and the difference between pseudoscience and actual science.